Hello, Moto.
Through this presentation, the learning objectives to be achieved are: firstly, describing the mechanism that leads to the development of Hashimoto's thyroiditis disease, and secondly, outlining the significance of understanding the implications of this disease. This butterfly-shaped gland in the lower region of your neck, when functioning normal, keeps everything in check. Should something arise and this thyroid gland becomes underactive, Hashimoto's maybe, but don't be presumptive. There are symptoms and signs that will let you know. Is it really hypothyroidism? Is that you, Hashimoto? These symptoms may be obvious, others not so much. Without further ado, let's list all symptoms out as such. If your neck is swollen or you have a goiter, difficulty concentrating and a hard time to ponder, constipation, dry skin, fatigue and hair loss, what about low tolerance to cold, irregular or heavy periods, or gaining weight just because? Shrunken thyroid later on that you're unable to see, but ask your doctor for a test to determine what it be. But how did this happen? Why me? Why me? Well, genetic or environmental factors both make this a possibility. Your T cells are helpful, the body's friend, up until the point that it attacks your self-antigens. In the case of Hashimoto's, your thyrocytes are attacked. There's infiltration of B cells and antibodies, all of which are facts. The helper subsets play a role in cytokine secretion, ultimately having a hand in thyrocyte destruction. The body tries through cell tolerance, T cell regulation, and the apoptosis pathway. The final lifeline may come in the form of immunotherapies, just in time to save the day. These therapies are centered around the use of iodine and through innovative methods involving synthetic thyroxine. Now we're at the end, and I hope this was fun, but please stay with us for the fun has just begun. This slide illustrates a flowchart which shows the factors involved that eventually lead to the development of Hashimoto's thyroiditis. So getting right into it, there are two main factors known to contribute to the breakdown of immune tolerance. These are environmental and genetic factors. Polymorphisms. When polymorphisms occur on genes that are specific to the thyroid gland, that individual is more likely to develop Hashimoto's thyroiditis. There are several SNPs that have been found to play a role in this disease. This diagram highlights exactly how these polymorphisms occur, moving from cytosine and guanine to tyrosine and adenine. Hashimoto's thyroiditis is more prevalent in females as opposed to males, and this could be accounted for by the X chromosome inactivation, where instead of having a normal 50-50 distribution of inactivation, there is distribution that favors either the maternal or paternal origin. This would result in one of these origins being expressed at significantly low levels, where the body may not recognize the antigens as their own. This diagram shows the difference between a normal distribution and an uneven distribution, also known as a skewed distribution.
So now we are going to look at environmental factors that contribute to increased incidence of Hashimoto's thyroiditis. Iodine intake. Increased ambient intake of iodine increases Hashimoto's thyroiditis. When there are excess levels, it inhibits and suppresses the body's ability to regulate and rid itself of dysfunctional cellular components via autophagy. All things are poison, and nothing is without poison. It is the dose that makes a thing not a poison. A quite fitting quote from Paracelsus himself. Studies show that there is a decrease in the levels of thyroid peroxidase antibodies in non-smokers, and this was supplemented further by studies that show there was an increase in the level of individuals developing Hashimoto's disease subsequent to quitting smoking. Despite having positive results in patients with Hashimoto's thyroiditis, smoking is not advised. Moderate consumption of alcohol has been proven to be beneficial in protecting against thyroiditis. The main takeaway from that is little can go a long way, with the key word being moderate. Selenium intake. Low intake of selenium is strongly related to the development of Hashimoto's thyroiditis. Taking selenium has shown to boost the immune system Vitamin D. Studies conducted tested for the influence of being deficient in vitamin D on Hashimoto's thyroiditis, and while the results appear promising, several factors such as BMI, age, estrogen use, and seasonal variations were not accounted for. Infections. Thyroid follicles in patients with Hashimoto's thyroiditis has MHC class 2, while patients without this disease does not have MHC class 2 on the surface of these thyroid follicles. The expression of those molecules could be induced by interferon gamma, which results in thyrocytes presenting either foreign antigens or self-antigens on the T cells, which leads to the activation, thereby initiating an autoimmune response. Stress influences the development of autoimmune diseases associated with the thyroid. When under stressful environments, the body produces hormones such as catecholamines and cortisol, which then inhibits the immune system by preventing production of white blood cells. Lastly, pregnancy. Since hypothyroidism is hereditary, it is possible that if a mother or partner makes a child with either parent having family who have Hashimoto's thyroiditis, it can result in the offspring developing the disease.
Pathogenesis, the breakdown of the immune system. This is caused by either the combination of individual factors or by a single individual factor leading to the immune tolerance. As we have already learned from the class, antigen-presenting cells, or APCs, take up antigens, process them into peptides, and form antigenic determinants, or epitopes, which are then bound to MHC class II molecules. The MHC class II molecules with the bound epitope are recognized by T cell receptors on a CD4+ cell, resulting in the activation of that CD4+ cell. Following this, proteins B71 and B72 on other antigen-presenting cells would bind to the protein CD28 on other T cells. This would then lead to the induction of cytotoxic T lymphocyte-associated protein 4, or CTLA4, which reduces the interaction between APC and the T cell, terminating the immune response. Self-tolerance, which occurs in the thymus, is known as central tolerance. Tolerance in peripheral tissues, however, are known as peripheral tolerance. The immature T cells that originate from the bone marrow enter the thymus where they undergo positive and negative selection, both of which we have already learnt in class. Central tolerance does not, however, guarantee that all reactive T cells are not selected. When a naive T cell interacts with an MHC class II molecule without the protein CD28 of that T cell undergoing ligation, anergy occurs. Anergy is the mechanism whereby a T cell undergoes functional inactivation when it meets an antigen. Regulatory T cells are also produced in the thymus, and they tend to live in the peripheral tissues in the event that any self-reactive T cells escaped central tolerance. These regulatory T cells produce IL-10 and TGF-beta, which directly inhibits T cells. A type of T helper cell, Th17, also works with the regulatory T cells to achieve immune homeostasis. Th17 cells in particular are heavily involved in inflammatory responses, in addition to being a link to Hashimoto's thyroiditis. This is an illustration showing how both central and peripheral tolerance affects autoimmunity in this particular disease. In Hashimoto's thyroiditis, there is an infiltration of B lymphocytes into the thyroid gland. Through the activity of Th1 cells, cytotoxic lymphocytes and macrophages become activated and orders the destruction of thyroid follicle cells. Activation of the Th2 cells, however, results in the stimulation of antibody-producing plasma cells. The subsets of the T cells involved in this autoimmune disease are outlined in the diagram seen, which shows what induces their differentiation and what cytokines they produce. The mechanism that occurs following the breakdown of tolerance leads to the destruction of the thyroid gland through autoimmune mediation of both the B and T cells, which is a characteristic feature of this disease. Additionally, thyroid peroxidase and thyroglobulin antibodies may evoke an antibody-dependent cell-mediated cytotoxicity, or ADCC, which involves activation of the complement system, where through a cascade mechanism there is a formation of a pore-forming complex known as a membrane attack complex, or MAC, that results in lysis of the thyrocytes. The humoral and cell-mediated responses of B and T cells, once signaled by the T helper cells, are neatly summarized in this diagram. So now we move on to the types of variants that exist within Hashimoto's thyroiditis. The goitrous variant. This was the first variant to be discovered by Dr. Hakaru Hashimoto in 1911. It has the characteristic development of a goiter. It has undefined lymphocytic accumulation within the structure, accompanied with a size that exists within the secondary lymphoid organs, which function as a region that allows growth and proliferation of B cells known as germinal centers. This diagram shows exactly how the goitrous variant looks in a histopathology. The Herzl cells are easily identifiable and is labeled to the left of the diagram. Looking closely, you can see that it has a pinkish appearance. Another distinguishable feature is a lymphoid follicle, which is the mass of tissue seen aggregated to the right of the diagram. Lastly, there are also silent and postpartum variants, hashitoxicosis, atrophic variant, IgG for related variants. Juvenile variant, as the name suggests, predominantly occurs in children. Juvenile variant.
So let's move on to immunotherapies for Hashimoto's thyroiditis. One immunotherapy method is the use of thyroid hormone replacements. This is the primary method used for treating patients with Hashimoto's thyroiditis. Here we have an illustration of the synthetic T4 tablets. Another immunotherapy method, which has newly emerged, uses thyroid. It is important to note that this form of immunotherapy has not been FDA approved. This flowchart illustrates the procedure to determine the correct method of immunotherapy. So this brings us to the end of our presentation. We hope that you all have learned something new today, and we are eager and willing to answer any questions that you all may have for us. Thank you for your time and for your attention.